Welcome back to the course on fluorescent spectroscopy. We're already up to section 8, and at this time we've been discussing fluorescence quenching. And notice this as part 8C because it's the third part. In the previous two parts we discussed the standard types of fluorescence quenching. You always read about static and dynamic and fluorophores and membranes and that type of thing. But as I was putting this together, I realized that there was sort of another class or multiple classes which were a different type of quenching that didn't fit cleanly into either of these parts. So I just decided to put them together in a not maybe a logical pattern, but a somewhat logical pattern, just to explain what's happening in each of these unique systems. And the first we're going to start with is a 4-helix bundle, which is a structural motif in many proteins. You can sometimes find these in natural pieces of proteins. Probably more often these days, these are designed so that by molecular modeling so that they will form a 4-helix bundle. Typically, they're held together by hydrophobic groups. So what happens in this case in water, which doesn't like the hydrophobic groups, the 4-helix bundle sticks together, and when it does, it leaves a hole in the middle for halothane, which is an anesthetic. And that anesthetic quenches the 4-helix bundle. What I failed to say was that this bundle is going to contain a couple tryptophan residues in it. And we're going to see what happens when we break apart these hydrophobic interactions. So this is really what it looks like. If we take a close-up of part of this 4-helix bundle, we see that it has a couple tryptophans in the middle. If it didn't, we wouldn't have anything to look at. I don't know why they marked them methionines. They're of no significance here. But the significant molecule is the halothane, which is this little molecule in the middle. And to my knowledge, halothane doesn't bind anything, so it must be rattling around in the cage. So one of the questions I had, and I've asked this to students at times, and I didn't know the answer, so of course the students know the answer. Is this static or dynamic quenching? Is this halothane staying in one position and just quenching the tryptophan, or is it bouncing around like it's in a cage? And I frankly don't know the answer to that. We don't know if it's a static or dynamic process, and we don't know if the halothane is trapped in that cage. Now we could design experiments to test which of those there are, but those weren't done in this original paper, so I can't give you the answer to that. But it's a type of quenching which is really created because the quencher is localized in a small volume around the tryptophans which are being quenched. Some of you might remember this slide from a previous lecture, and at that time you probably wondered why I was showing it to you. Guess we've just changed topics. It was indole or tryptophan being quenched by either protonated imidazole, which is imidazole-M, or by the neutral version, which is just imidazole. And what was found was only the protonated form was the quencher, and that's because it was an electron acceptor. Indole likes to donate electrons in the excited state, so it's a photo-induced electron transfer. This might seem far removed from many proteins, but I stumbled on what I thought was a beautiful example of where these types of interactions played a role in the protein structure itself. These are peptides from an HIV protein called VPR, which contributes to the virus life cycle and the high morbidity and mortality of AIDS. And it seems to have histidines and tryptophanes, which are located close enough to one another that they might actually bind with another, and that might be the one of the factors that holds these two helices together. So this was tested. It's known that the unfolded protein has a number of helices on it, but we're only dealing with the two strands that are the same but moving in opposite directions. And it seemed probable that positively charged histidines would be in some type of complex with the indole residues or quenching the indole residues. So this seemed likely to be a case of another case of photoinduced electron transfer. So how was this tested? This was tested by looking at the emission spectra of this dimer of helices and how it was affected by pH. So this is first what we would call the wild type. It's really fragments of a wild type protein. And as the pH dropped, the quenching got less and less. And going to this region here is we're going from the region from the neutral imidazole group down to the positively charged group, and the positively charged group is the quencher. So that pretty much confirms what's going on in this duplex. And a similar thing was found with the version containing only one histidine, but the results were not as dramatic. This is a very telling curve because it's looking at the tryptophan fluorescence as a function of pH. So as we start at high pH, the tryptophan fluorescence is pretty high because there's nothing to quench it. The imidazoles are all unprotonated. As we drop through the pKa of protonating the imidazole, then the fluorescence goes down. So I think this proves that these types of interactions can occur in model systems and actually in real proteins. Changing topics again, I'd like to talk about GFPs and how they can actually become sensitive or as sensors. In general, the GFPs display a fluorescence which is really not sensitive to the outside environment at all. The fluorophores and the GFP are locked in this beta barrel. Usually the beta barrel protects the fluorophore completely, and it displays pretty much the same spectral properties wherever it is, similar to the lanthanides we described earlier, where the fluorescent orbitals were shielded by an outer layer of s electrons. But this particular one is responsive to both pH and to chloride. And this was sort of unusual because we don't really have a good reason why both should affect it. We can imagine that if it's sensitive to chloride, it would be quenched by increasing amounts of chloride. We didn't have a good reason for why it should be quenched more at a lower pH value. So the authors figured this out, and it's a pretty interesting explanation. It turned out, well, as I said many times previously, static quenching is expected to change the absorption spectra of the fluorophore. Dynamic quenching or collisional quenching, is not expected to have any effect on the chromophore. So here we see that the presence of chloride actually changes the absorption spectrum of the chromophore, so that means we're changing the structure of the chromophore. So this quenching is really not static nor dynamic. Now the way that the quenching, the charge changes as we go to the higher, is going to be the higher or the lower pH values. We'll figure that out in a second. As the pH changes, more of the charge of the chromophore and the GFP is moved to the outer part, which is more exposed to the surface. That outer part is going to be repulsive to the negative chloride, so it's going to decrease the amount of chloride quenching. So this is a combination of effects that it is affecting the chloride. So the reason for less quenching at the high pH is really the negative charge, which may repel a chloride from coming in. And these wonderful little proteins also respond when they're in cells. Probably going to be more difficult to use than a mono analyte sensitive GFP, which is not going to have this more complex behavior. Molecular beacons. I have to talk about molecular beacons because they're such a major tool in biotechnology and drug discovery and many other things. The basic idea of a molecular beacon is to have two regions with a fluorophore and a quencher, or fluorophore and a receptor, and they are bound together by a loop sequence. And the loop sequence is the thing that we're looking for. So we go get our DNA sample from Jurassic Park. We're looking for certain sequence that occurs in Tyrannosaurus rex. You probably have to amplify the DNA, but if we amplify it enough, we make the complementary sequence to the loop sequence, and that moves the fluorophore and the quencher away from one another. When this first came out, I thought, boy, this is such a simple idea. Anyone could do this. And it sort of proves how narrow-minded one can be in fluorescence, because I didn't understand the major impact this simple discovery was going to have. So right now there's many types of different PCR methods. Some of them disrupt and the fluorophore and their quencher, the fluorophore and the acceptor, which can be monitored by changes in the donor emission. There's reverse transcription PCR. We're using RNA as a starting material rather than DNA. These are done in multiplex reactions, and these are even done in microdots, where you have microdots of an aqueous fluid flowing in a sort of a fluid non-aqueous media, and you have individual PCR reactions going on in each of these microdots. And when you make them small enough, you probably only wind up with one piece of DNA in the microdot. So the result is really a consequence of one specific sequence. When this first started, the molecular beacons originally contained fluorescent donors and acceptors. So the first one looked something like this, had a CY3 on one end, and a CY5 on the other, I believe. Again, this is the sequence we're testing for, and as you add this sequence to the sample, it breaks apart. This becomes stiff because single-stranded nucleic acids are much more flexible than double-stranded nucleic acid. breaks this apart, see much more of one fluorescence than the other. So one goes up and the other goes down, and you can tell that you have identified the sequence that you're looking for. One of the problems with these early measurements was that there was always a desire to do multiplex measurements, and the emission spectra could overlap. So, especially on the long wavelength side. So if you were trying to measure emission on the red wavelength side, most fluorophores have a longer tail on the red wavelength side, so you're going to get some contribution from the shorter wavelength fluorescence, and that was a nuisance, and the scientists wanted to get away from that. So they came up with a rather ingenious idea. Instead of using a fluorophore at the other end, they used something called dapsol. And dapsol was coined as a black hole receptor. It absorbs the energy from the fluorescent group, but it does not display any emission. And for most PCR experiments, all you really need is the fluorescence from the donor part, and the acceptor part only gets in the way. So this was I believe, the first black hole quencher that was reported, which was a dapsol, has this azo group in the middle here, and the emission spectra looked like this. This was the Danzel absorption spectra from this. I'm going to call it Danzel, even though they call it E-Dans there. It looks like Danzel. We have its emission there, and here's the black hole quencher overlapping with the emission. So you're going to knock out that emission and only see the Danzel emission. That worked fine, except that there was a desire to use the molecular beacons for more types of assays, especially multiplex assays. So while the dapsol worked with the short wavelength emitting Danzel type group, it didn't work with longer wavelength emitting fluorophores. And these are some of the standard fluorophores used in DNA analysis. So there was an effort to make black hole quenchers that absorbed at longer wavelengths and would therefore get rid of the emission from these other species. And I just happen to like this diagram. I know it's similar in content to the previous one, but it sort of explains where we were with black hole quenchers. When DNA sequencing was just getting started, these were the absolute favorite four fluorophores that were used for DNA sequencing. And this first black hole quencher, with the yellow here, hard to see, was not very good for that. So other black hole quenchers had to be made to deal with the sequencing probes. And for a while, they didn't have the black hole quenchers, so they actually used long wavelength CY5 dyes to be the acceptors and filtered out their emission. But that was quickly corrected, and black hole quenchers have been made for just about any wavelength region. And there are efforts to make the region of the black hole quencher wider and wider. I had a couple of papers on that, but they were complex to present, and the basic idea was to design a black hole quencher with multiple transition moments in it. But anyway, let's look at the quenching one. Everyone has an azo group in it. So I guess that's the secret for making a black hole quencher. And you can get it to work at long wavelengths or shorter wavelengths. And a lot of the names overlap. You can find the same black hole quencher with a different name from a different vendor. So it's easy to look for the spectral properties. It's not always easy to sort out the names. The mechanism by which a black hole quencher works is not easy to know, and this is for the following reason. If the donor and the acceptor or quencher molecules are far away, you expect RET to be occurring. So there would be fluorescence resonance energy transfer. Now, if you have fluorescence resonance energy transfer, you always want to have some donor emission as well as acceptor emission, or even if you had a black hole quencher, you're going to have some donor emission. So what you really want is to have no acceptor emission at all, and that means there's a tendency to try to put the donor and the black hole quencher close together. And some groups will claim that the mechanism of quenching is that these two groups stack together, that is the chromophore and the quencher, and it's that complex that's non-fluorescent. But if you just look at the rules for energy transfer, it's going to be very rapid if they're that close together. So I don't know which one is true, whether it's the quenchers are due to rapid energy transfer, or if they're forming actual static complexes. Here's a quencher you probably don't know about. If you happen to have a kilobar gold sitting at home, well, the first thing I'd advise you to do is to quit your job and go buy a Lamborghini or something. But there are other uses for gold, and those are gold nanoparticles. And the gold nanoparticles, first of all, they don't cost hardly anything because they use so little gold. But they can be made to have only almost any absorption spectrum that you would like. And gold is really completely non-fluorescent. So it's just like a black hole quencher. There are some old-time reports of saying there was some emission from gold, but the quantum yield had 4 zeros in front of it or something, so you didn't know what to make of it. So you can get almost any absorption spectrum you want, and there's all types of gold nanoparticles. Some are nanospheres, some are nanorods. Almost any of those can be used for a black hole type experiment. And here's one of the first molecular beacons that was based on gold nanoparticles. So they had a DNA sequence that they bound to the gold with a sulfhydryl group. There are many ways that you can bind things to gold, but the sulfhydryl group is always the easiest. Since the strand to detect the target sequence was single-stranded, it was always flexible, and for unknown reasons, the fluorophore, which was tetramethylrhodamine, would spontaneously absorb to the gold and would be completely quenched. So you have a black hole. You have a molecule that doesn't fluoresce at all unless you put some of the target DNA in it. If you add the target DNA, that changes the single-stranded DNA into double-stranded DNA. That's a lot more stiff. It's stiff enough to yank the tetramethylrhodamine off of the gold, and it becomes fluorescent. So I think this is a great way to make molecular beacons. This is also being done on surfaces where you spot the surfaces with different sequences and use an imaging methodology to figure out which target DNAs are present. The next slide is really one of my favorite slides. It's something I always wanted to do, but I didn't know how, and I can kick myself in the butt for not figuring out how to do it. I always thought it'd be good if somehow we could amplify fluorescence rather than quench fluorescence. I know this is sort of seems out of sequence for a quenching lecture, but I'll mention it anyway because it actually was a quenching experiment. The what was done here was they compared the quenching of the monomer of this particular polymer, which I believe is called PPV, and the other side part is for the side chains that are on it. And if we compare these two, that we're going to use them as our starting molecules in a quenching experiment, well, you can get almost nothing from the still beam, but if you have 1,000 still beams in one place, well, maybe you can get a pretty good signal, the orders of magnitude larger than the single fluorophore. If you try to do quenching of still beam by methyl-viologen, which is the electron acceptor for still beam, it has a very small amount of quenching. Quenching constant is approximately 15 mole to the -1, which means you need 0.07 molar methyl-viologen to get 50% quenching. So that's not very effective at all. It's not useful. And anyway, there's not enough of a signal out of the still beam. The brilliant part of the experiment was using this long chain that had thousands of still beams on it and quenching it with methyl-viologen. And I don't think I would have expected this result, but binding just one methyl-viologen to this chain can quench 1,000 monomer units. It turns out, stating in another way, it takes around, 650 femtoseconds for an excitation to move all the way through the chain. And if there's a methyl-viologen anywhere near that on the chain, it's going to quench the whole chain, which only had a single excited fluorophore in it. So basically, the quenching is about 1,000,000-fold more effective. I didn't show you the other part of this experiment where they developed the chemistry for removing the methyl-viologen to turning it into an assay. But the interesting part is what I would call amplified quenching. This might be getting a little bit complicated, so I wanted to show you reasons why this particular polymer is useful for you. This polymer makes the whites whiter. If you have this polymer in your washing solution, it's going to be whiter with the polymer. If you have a grody pair of jogging shoes, they're going to look whiter. And this is actually an illusion. The way these really work is many of the detergents that are used contain these types of polymers, not necessarily this particular structure, but this type of polymer. And if you go to a good party where they have a lot of dark lights, you'll notice all of the white clothing is glowing blue. And that's exactly the purpose of this polymer, because the blue light coming from your clothing when you're out in the sunlight counteracts the yellow of your clothing that is accumulated over with time, and your whites look whiter. So this is a real world application. I would guess if you get spots of methyl-viologen on this, it's going to turn dark, but how's that going to happen? And there are various types of polymers along these lines that can give different color luminescence. I don't know how my shirt is showing up on this, but I noticed when I first looked at this shirt earlier when I was starting the lecture, I noticed that it looked more blue than usual, and I thought that was because of the way I positioned light on the desk that gave this shirt a bluish tinge. Well, anyway, that's the end of part C. I hope to end it on sort of both a practical note and a humorous note, I hope you enjoyed the lectures. That's everything for quenching for now, and we hope to see you at a subsequent lecture. Goodbye for now.